uh, together with Alina and Steve. So to basically give what I would be talking to you about would be basically these 4 important steps. And so how usually aging clocks are made is that you have some sort of biological data, usually from biomarkers, that goes in data pre-processing, as we saw in the keynote before, that how important is that. Then usually we have kind of a feature selection step, which we will see also why is it so important for these prediction models. Then we basically have a kind of a testing approach where you do cross validations over your training data. And then you basically use a kind of model to model the information that you want to use for prediction. And lastly, I would also talk about a little bit about interpretation of the model, which I think makes things more biologically meaningful. Okay, so pre-processing, actually what we implied over here was pretty standard. We used the CHAMP directory. So basically CHAMP is a standard DNA methylation processing pipeline. And it was also implied, it was also recommended by the organizers. And so we use actually a bunch of steps from the pipeline to filter out kind of a quality control CpG probes, which can be problematic for the model and to make the model more robust. We also perform normalization for the type 1 and type 2 Illumina probes to remove the bias using the BMIQ method. And something that we found out that our data was confounded with batches, but since the information was not really available for the test data, we rather chose to skip it, but we could find that there were batches in the data which we can potentially correct for, but we actually do not use it finally. So even before going deep in our strategies and approach, this is a simple PCA plot from all the CpGs that we get after filtering step and normalization. And what you see over here is that you basically don't see any gestational aging pattern. So samples don't really cluster very nicely. And the idea is to basically develop this feature selection step in a way that we can help improve this signal of gestational age from here on. And to get that, what we did first was to do kind of feature selection step where we select for those CpGs which kind of correlate very good with age. So we selected 4,000 CpGs from Spearman's correlation, a bunch of them which were positively correlated and bunch which were negatively correlated in the two extremes. And this is how they look like. So I hope what you can appreciate on this heat map is that each row is a CpG, and from left to right, you have samples arranged from young to old. And what you can see that there are a bunch of CpGs which were very highly methylated, or they were not methylated before and they gain methylation. And then there were CpGs which were kind of non-methylated before and gain methylation, and which were methylated before and lose methylation. So it was a clear demonstration that we do have trends in our data, and the idea was to basically capture it. What we did find during our approach that to move ahead, instead of using these CpGs directly in the model, we can actually do kind of a feature engineering step where we kind of modify these features. So the CpGs, how usually these aging predictors are made are through these kind of regression models where you have CpGs directly inputted as features to the model. But what you can imagine, a potential problem here is that the models are kind of biased towards each CpG because the model themselves contains CpG values, which can be affected by things not pertaining to gestational age. So for example, technical bias, which can change the value of certain CpG, which can then create a problem for prediction outcome from the model. To basically circumvent this, what we kind of developed and incorporated here was a clustering approach. So basically, we take these CpGs, as I showed you in the last heat map, We cluster them using an unsupervised clustering method, and then we use rather the clusters as features to the model. And what we found out that just by doing this additional step could could make the models more robust and remove the bias of individual CpGs in the model, and it basically performed better in our hand. And now coming back to the PCA that I showed you before, what you can hopefully appreciate here is that after doing these couple of steps of correlation filtering and clustering, we could very now nicely separate these samples on their gestational age, which was not possible before. So it was kind of nice for us to see that the approach works in a way that we could pull out the gestational aging signatures from these CpGs in a better way. Next things, so now to test our modeling approach, we wanted to see how the data was distributed. So it's a simple on the x-axis, we have all the studies with their age distribution. And what we did was to kind of design an approach where we wanted to have this kind of a leave one out cross-validation approach. So we categorized the studies in 3 age groups. And the idea was to basically exclude one study from each age group in each iteration of cross-validation or fold. So for example, here as you can see, so this study, and this study would form the test set, and everything else goes in the training of the model. And then in the next iteration, we just replace this study with this another study from the same category of age group, and we again do the modeling. So by this, we could basically we could design a more robust cross-validation approach to test how our modeling in general performed, and how the feature selection and all of these things affect the performance. And now to coming to the results a little bit, we implied a rather simple modeling approach. We used elastic net regression, which is kind of very common, and it's also relatively simpler model to interpret. Yeah, so elastic net regression with the penalties. And what we already see here is on the x-axis, we have the actual age of the samples, and on the y, you have the predicted age. And the model already performs pretty accurately, so the colors are different data sets. But you do see that there are a bunch of these samples which are predicted, like they're kind of off from the prediction. So which we thought might be because these are kind of disease samples or outliers for our model. And so one of the ideas was to also design one more model where we train a final model by removing these samples from the training data set, and then maybe we get a more robust model which is not affected by these outliers. And good part about all of this was biological interpretability, so which I just briefly mentioned in the beginning. So the CpGs, essentially, they depending on where they're positioned, especially in the regulatory elements such as enhancers and promoters, they might influence the gene expression, what we know kind of. And so basically, my idea was to find that these CpGs which we train on in these models, what genes are these related to, and to basically narrow down on a bunch of CpGs to look for their biological significance. I kind of looked at the overlap between 2 models. So the model that I showed you before, and another very similar model, just with the PCR removed outliers being removed. And what you can appreciate is that we had like about 248 of these CpGs, which were common between these 2 models, which were very, very similar in their approach, just that one model, the second model, does not have a bunch of outlier samples. With these common set of CpGs, I wanted to do kind of a biological analysis, and to associate them with genes. We could already see that a lot of immune-related genes popped up in our analysis. So for example, so a bunch of these genes are related with leukocyte differentiation, lymphocyte differentiation, immunity, innate immunity, GTPase activity, and so on. So basically, the idea is that what these CpGs, which we train in our model, they kind of indicate towards those genes which are involved in immune-related processes, which is kind of interesting because what we also found was that there was a paper in 2017 where the authors trained a clock on from the blood samples of mothers. And what they found out was that there are a bunch of immune-related processes which are changing, and you can actually kind of you can build a model on these changing factors in bloods uh, of, uh, of mothers during pregnancy, and you can nicely track the age during pregnancy. And a good overlap that we find is that there are a bunch of players from their clock model, for example, the STAT5, and a bunch of players from our findings, for example, this JAK, which kind of work in a similar fashion. So this JAK STAT pathways is a very well-known signaling pathway. So it was nice that we also get our validation from already published literature, and it's also nice that we tap into biology as well, along with having an accurate predicting model. And so happily we submitted this model to the leaderboard, and we already got a nice result. We got a nice RMSE, so we were quite happy. But then because we were also doing this another parallel approach with the sub-challenge 2 from 850K array. The last model that I submitted here performed slightly better than our the other model that I talked to you about. So I mean, it would have not make a lot of difference for us in terms of performance in this dream challenge, because we would have still been second with our other model if it was still the best performing model. But irrespective of that, the second model actually performs slightly better. So just to also give you a summary about what this model is. So it's also very similar actually. It's just that now things are from 850K data, and we have a slightly different feature selection step. So if you remember in the first slide, as I talked about, this is a very important step during prediction modeling. And here the difference is that along with the correlation filtering that I was doing before, we also add this EVA's CpGs for gestational age from this Lee paper. So essentially, this is also kind of a fancy correlation filter that they incorporated in their EVAs to find the EVA's CpGs, which are kind of getting out from these weighted co-expression gene networks. And so basically, what here I'm doing is I take those CpGs which are these EVAs correlated, and together with the CpGs that I get from the correlation filter, we take all of them in the model. And another interesting thing was that here we don't perform clustering. So yeah, so like kind of couple of changes we had in this approach, but with a very similar elastic net regression. Still, we submitted this model, which is essentially this one, which performs the best on test data. Yeah, so this model, which does perform the best on the test data, did not really perform better for us in our hands on the cross-validation data. So this was kind of surprising, because the values that we get here for our cross-validation is actually kind of worse for this model than the model that I showed you before. And this was the rationale why we submitted the other model as our best performing model. And this is also something that I want to highlight over here, is that these challenges are kind of critical when we talk about performance over this test data, because in our hand, in our cross-validation, the other model, this sub-challenge 1 version 1.0 model performed the best, but it kind of, the RMSE really fell to, I mean, it was still similar for that the sub-challenge 1 model performed better in the leaderboard, but then on the test data it was completely switched. So now we get this other model from sub-challenge 2 perform slightly better. So I mean, this is kind of we don't really have a very definite answer to why this happens, but we speculate because there are, as we know, different populations in the test on the leaderboard and on the test samples, and also these kind of disease samples affect the performance of the models, and this might be one of the reasons why this discrepancy happens. And to conclude everything, we basically were able to develop approach by using filtering and clustering CpGs, which kind of very comprehensively capture the gestational aging pattern. We could also find that these common CpGs relate to immune-related processes in placenta, which is interesting because it is also similarly what others have found during which others have found to change during pregnancy. Of course, there exists a discrepancy over some over cross-validation that we designed leaderboard and the test data, and this is something which we should kind of figure out now, that what probably a more robust method of testing it. What we also could do is that we found out that there are batch effects in the data, as usually they are with the biological data sets, and potentially we can correct for them to improve the performance of the model. We also found this out during our approach, but since the information was not available on the test data, we did not do it. But again, this is something which can be incorporated now when we want to take this ahead. And also, we can improve the prediction accuracy through epic arrays, which I also found out that we get more correlated CpGs from this 850K array, which basically have a better resolution of the genome. But the challenge always remains in either using this smaller sample size or using CpG information or imputed CpG information, because one kind of, so using a smaller sample size hampers your power of the model, and using imputed information kind of also is problematic, can be problematic. So with that, I would like to acknowledge everyone in this project, especially Steve and Alina. We were working together on developing this approach. My home institute, Leibniz Institute on Aging, the graduate schools I'm being part of, and everyone from here, from the challenge, Professor Addy, we have exchanged a lot of emails, and thanks for the organizers for making this happen, and for making me travel all the way from Germany to here to present it. And this is the QR with all of our approach, and yeah, and if you are interested in, you can scan it, and you will also get the methods in more detail, and all the git codes if you are interested. Thank you.
So the question is, was that overlap of that kind of enrichment significant, or it just how likely was that to happen by chance?
So to perform the enrichment analysis, I made sure that we had a proper background of the genes. So yeah, I mean, before doing the enrichment, we also take into account proper background. So for example, the genes which are only expressed in placenta, so that we just by chance we do not, we don't find these genes which are related to placental processes.
So you take the set of genes associated with gestational age maternal blood, and consider it as a set, and you find an enrichment for this set among those that you have association with methylation, right? That's what you're saying. It just happened that some of the genes were in common.
Yeah, no, so it just happened that some of the genes were in common. Yeah. Yes, absolutely.
surprised that your performance in a test set swap with the cross validation, right? But did you make any estimate of the statistical significance of that change?
You mean the differences that we find between cross validation and test data sets?
So the error bar between one of the models and the other model in the leaderboard, those are numbers that have error bars.
Yes.
Are those error bars big enough to explain this as not significant?
I mean, well, I did not look at that, but error bars I don't understand, because so these are like really like one RMSE values for that we have from the leaderboard. So basically one RMSE value for the model that we get from the leaderboard. The error bars would be here for.
For example, we do typically bootstrap.
Yeah.
Do we have different RMSEs?
Yes, for different folds of, yeah. So for our data, we did that definitely. For cross validation, we had that, but we don't have that for the leaderboard and test. But maybe with together, we can definitely look for that, because yeah, I think it would be interesting, because we see this kind of drop from our cross validation to the leaderboard, and then kind of also differences between leaderboard and test. So I think, yeah, it would be really nice to look at that.